Alrighty guys, today we're going to do a karyotyping assignment. So if you go into the assignment tab, it says complete the following web lab on karyotyping. All the information is there for you, and if you get stuck, you can use the video, which I'm making now, for help and guidance. If your browser won't open the link, please follow along with this video, and you'll be able to get all your answers that way as well, because I'm actually going to go through the lab step by step. So it says this tool needs to be loaded in a new browser, so you'll click on that and it should pop open with a new Google doc window. I'm going to use mine in a Word document, which I have right over here, just because it's a little easier for me to type it on here. Then I don't have to go back through and erase everything. So you're going to be going to one website and you have to answer questions about 3 patients. So you can see it's only 21 questions long. And this is the website. If you click on this link here, this is what it's going to take you to. It says Karyotyping Activity, the Biology Project, Human Biology. It also has instructions in Spanish if you'd rather look at the Spanish version. So the first 5 questions you're going to be answering based upon the reading here. So let's see, it says, how are the karyotype analyses conducted? So you have to go through, we'll read through that. This exercise is a simulation of human karyotyping using digital images of chromosomes from actual human genetic studies. You will be arranging chromosomes into a completed karyotype and interpreting your findings just as if you were working in a genetic analysis program at a hospital or clinic. Karyotype analyses are performed over 400,000 times per year in the United States and Canada. Imagine that you were performing these analyses for real people and that your conclusions would drastically affect their lives. During mitosis, the 23 pairs of chromosomes condense and are visible with a light microscope. A karyotype analysis usually involves blocking cells in mitosis and staining the condensed chromosomes with germicide dye. The dye stains regions of chromosomes that are rich in the base pairs A, which is adenine, and T, thymine, producing a dark band. A common misconception is that the bands represent single genes, but, in fact, the thinnest bands contain over 1 million base pairs and potentially hundreds of genes. For example, the size of one band is about equal to the entire genetic chromosome for one bacterium. So that's kind of crazy. So when you're looking at these bands here, it's not showing you just one individual gene. This is just a place where there are genes located. So how are the analyses conducted? Let's see, it looks like we're getting to it here. We take a picture and then you stain the cells or stain the genes. The analysis involves comparing the chromosomes for their length, the placement of centromeres, which is areas where 2 chromatids are joined, it's that middle portion, and the location of the sizes of G bands. You will electronically complete the karyotype for 3 individuals and look for abnormalities that could explain the phenotype. So how are they conducted? Well, I'm going to go back up to this paragraph here. Talks about, you take a picture and stain the regions of chromosomes that are rich, I'm using this sentence here, that are rich in base pairs, producing a dark band. What are some uses of analyzing karyotypes? Well, we know that we're going to be looking for abnormalities. You could also use this for paternity testing. And they also use this in like CSI stuff. What causes a dark band on the chromosome? The base pairs A and T. Anytime they're located together, you'll see a dark band. What is a centromere? Let's see, where did we have that? That would also have been part of our notes, but if you don't remember it, here it is, centromeres, areas where the 2 chromatids are joined. Which chromosome determines sex in humans? And what is an autosome? the X and Y determine sex in humans. And an autosome, now is this an autosome or sex chromosome? It's a sex chromosome. We know that because, well, it's determining the sex of the organism. An autosome is all the other ones. Okay, so now we're going to click on patient histories. Well, I guess I could read the assignment part first. This exercise is designed as an introduction on genetic studies on humans. Karyotyping is one of the many techniques that allow us to look for several thousand possible genetic diseases in humans. We evaluate 3 patients' case histories, complete their karyotypes, and diagnose any missing or extra chromosomes. Then you'll conduct research on the internet to find websites that cover some aspects of human genetics. All right. So we're just going to do the patient history part of it. We're not really going to be doing the other web part of it. So I'll click on patient history. Here we have patient A. And our instructions here, it says match the chromosome to its homolog, which is its matching pair. After all the matches are complete, you analyze the patient, scroll down to view your completed karyotype. So what is patient A's history? Patient A is nearly full term. Patient A is the nearly full-term fetus of a 40-year-old female. Chromosomes were obtained from the fetal epithelial cells acquired through amniocentesis. So the history for patient A, it's a full-term fetus. That's what they call a baby before it's born. Okay, so I'm going to click on the karyotype then. And again, if this doesn't work for you, then you'll just follow along with the video. So what we're doing, they give us a chromosome here. It says place the chromosome in the partially completed karyotype below by clicking on its homologous chromosomes. If you match the chromosome correctly, you'll proceed on to the next one. If you match it incorrectly, a page will explain why the chromosome you chose is not the unknown's pair, and you can choose again. So it gives you different ones you can pick from. They're highlighted in blue, so I can't pick the ones that are blacked out already. So I'm looking at this chromosome here, and I have to find a match for it. Well, it's a longer one, so I know it's not going to be 12 or 16. I'm going to go with 2. Let's see if it matches with 2. And it does. I was correct. Now I have to find this chromosome. Looks like it'd be little like that. Maybe that one. And that one. Now you've paired up all the chromosomes. Where does the extra chromosome belong? So it gave me 4 new choices. It's a little one. I'm going to go down here. All right. So I was correct. Congratulations, you successfully completed patient A's karyotype. Next, interpret the karyotype and make a diagnosis of patient A's completed karyotype at the bottom of the page for reference. Okay, I'm going to move myself up here so that we can read everything on this. How many total chromosomes are on your karyotype? So here's my karyotype. I have the 23 pairs, which gives me 46, and I have 3 on 1, so that's going to give me 47. What sex chromosomes does your patient have? So here's my sex chromosomes. They're either XX or XY. If it is XX, they're both going to be the same size. If it's XY, it's going to be a really long one matched with something that's around the size of 22 or 21. So this is going to be XX, so this is going to be female. Which chromosome set has an extra one? That's going to be number 21. What diagnosis would you give this patient? Okay. So if I look up here, let's read through this portion right here. Lab technicians compile karyotypes and then use a specific notation to characterize the karyotype. This notation includes the number of chromosomes, the sex chromosomes, and any extra or missing autosomal chromosomes. For example, 47XY plus 18 indicates that the patient has 47 chromosomes, is a male, and the extra number is on chromosome 18. 46XX is a female with a normal number of chromosomes. 47XXY is a patient with an extra sex chromosome. So for this particular patient, I would have done 47XX plus 21. The next step is to either diagnose or rule out a chromosomal abnormality in a patient with a normal number of chromosomes. Each pair will only have 2. Having an extra or missing one renders a fetus inviable. In cases where a fetus makes it to term, there are unique clinical features depending on the chromosome affected. Listed below are some syndromes caused by abnormal number of chromosomes. So we have an extra one on number 21. This is normal, Klinefelter's, Downs, and trisomy 13. So this is what we could potentially have wrong with this fetus. So if we read through the chromosome abnormality, it says the patient's problems are due to something other than an abnormal number of chromosomes. 1 or more extra sex chromosomes, which we don't have. We only have 2 of those. Trisomy 21, an extra chromosome on 21, we do have that. We don't have an extra chromosome on 13, so I'm going to go with Downs for this one. And then we're going to click on to patient B. Move this out a little bit. Okay, so we're looking at this little guy here. That looks like number 21. That one, oh, it's not chromosome 10. It's deceivingly similar. So I'm going to go click on try again. Let's go with 11. 14 maybe. Oh, it's over there, the sex chromosomes. That one looks like that one. This one, that one. Last chromosome, obviously all the chromosomes are already paired up, so this is an extra one. Doesn't look like 21. Maybe it's 20? No. Ah, okay, it was over on that one. Congratulations, you successfully completed patient B. Okay, so I didn't do their history yet, so I'm going to skip that question. How many total chromosomes are in my karyotype? Well, I have my 23 pairs of chromosomes, plus I have an extra one over there. So I have 47. What sex chromosomes does your patient have? You have XXY. Which chromosome set has an extra pair? It's going to be pair 23. Finish the notation. So if we go back up here, it tells us how to do that notation. So we have 47XXY. What is the diagnosis? Whenever you have an extra sex chromosome, that is going to be Klinefelter syndrome. Now let me go back to patient histories real quick here. Okay, so patient B is a 28-year-old man who is trying to identify the cause of his infertility. Chromosomes were obtained from a nucleated cell in the patient's blood. So I'll go back up here to my history. 28-year-old male looking for infertility reasoning. And patient C. Patient C died shortly after birth with a multitude of abnormalities, including polydactyly and a cleft lip. Chromosomes were obtained from a tissue sample. All right, so died young with multiple anomalies. I'm going to click on this one here. Put it over a little bit so I can see what I'm looking for. I'll go with 12. This chromosome, maybe number 3. 11. I feel like I'm going to get this one wrong. Put it over here. Okay. Almost done. What about that one's going to go down here? One left. 13. Okay. So, how many chromosomes are there in the karyotype? We have everything is all paired up here, except for number 13 has an extra one. So once again, we have 47 sex chromosomes. They have an X and a Y. It's on set 13. Write out the correct notation for this karyotype. So we got 47 xy plus 13. And the diagnosis here would be trisomy 13. Most babies do die if they have that abnormality. And that's it for this assignment here. Make sure you go back through and submit that. If you have any questions, maybe you were watching the video, it would help you out. But that's all I have for you today, guys. Have a good one.